Okay, so I want to welcome everyone to the 2022 Cell Gene meeting on the Mesa hybrid version. So those of us in the room in Carlsbad and those of us online as well. It's my pleasure to be the moderator for this first session, this workshop entitled Secrets of Scale: How the Convergence of Systems Biology, Engineering, and Nonconventional Regulatory Approaches to Accelerate Cell Therapies to Market. So I'm the Chief Regulatory Officer of the Advanced Regenerative Manufacturing Institute. And our main program is BioFab USA, which is a Manufacturing USA institute. I'm an immunopathologist by training and came to ARMI soon after they were funded by the DoD from the FDA, having spent almost 20 years working with cell and gene therapies, thinking about policy. I'll take just a minute to introduce Manufacturing Institute, ARMI, and BioFab, and what we are doing and how we got started. And then what I'm going to ask my panelists to each introduce themselves and speak for a short introduction of why they happen to be here on this panel. I know why they're here on this panel, and I think it's an amazing panel that represents lots of different perspectives on secrets of scale. They all know part of the secrets, so we're going to reveal non-proprietary information, but secrets that they have learned from their various perspectives and experiences. So BioFab, the idea that grew into BioFab thanks to the DoD funding and to the Manufacturing USA program in 2016, really originated around a table at a group of government agencies collectively known as MATES. That's a group that's been meeting for almost 25 years of folks from NIH, NSF, FDA, NIST, DoD, VA, NASA occasionally, over that period of time, who are interested in what we now call regenerative medicine, which when it started we called tissue engineering. So it has outlived several overarching names for the field. About 2014, 2015, we were looking back at our 2007 strategic plan for what we called tissue sciences and engineering at the time, and said, what we done that was been important in that field, but what do we have remaining? The thing that ended up that we realized that we had not made as much progress as we wanted to was scaling up manufacturing. How do you get from a very bespoke artisanal product to something that really could make a difference in the clinic? And we were talking about how we were going to solve that, and the opportunity to put an RFP for a manufacturing institute came up. We took advantage of it, and BioFab and Nimble both kind of came out of that effort across the government, which is my segue to introduce Lloyd Rose from the Department of Defense, who is our program manager at BioFab. Give a little background on Lloyd's background and where you fit into this world. Lloyd?
All right, thanks very much, Richard. So my name is Lloyd Rose. I'm a microbiologist by training, but after graduate school, I did my postdoctoral fellowship at the U.S. Army Institute of Surgical Research, and that was my introduction into Army medicine. And from that point forward, I have been working in regenerative medicine space, either in an administrative capacity or in research. And specifically, my research was focused on skin, burn injuries, and scarring, and how to ameliorate scarring following burn injuries in particular. And that's a big focus of the research at the U.S. Army Institute of Surgical Research, which is the Brooke Army Medical Center has the Army's primary level 1 trauma center for burns. But after that postdoc, I moved up to the U.S. Army Medical Research and Development Command, which is the organization that is the primary funder for Army medical research. And at that point, I broadened my experience into all the regenerative medicine focus areas that the Army had an interest in. And so beyond skin, that became skeletal muscle, peripheral nerves, as well. It continued to be skin, that did not change. Bone, have been the primary funder of vascularized composite allotransplantation. So all of these sorts of things that fall within the regenerative medicine spectrum. And one of the primary efforts that we supported during that time was the Armed Forces Institute of Regenerative Medicine, which was multiple consortia over many years, starting in 2008. And those amounted to several $100 million worth of research into regenerative medicine space. And the goal of those efforts was to treat, of the very first consortiums, was to treat one patient in one clinical trial. We thought that was a reasonable thing to do considering the state of the field at that time. And actually we ended up funding numerous clinical trials and treating numerous patients. Some of those things worked, some of those things did not work so well. But one thing that was very clear coming out of that was the degree to which the roadblocks that were identified in trying to bring these things to the clinic and then to bring them to the commercial marketplace. And it was pretty clear that figuring out how to manufacture these at scale was going to be one of the major roadblocks. Funding, of course, is always an issue, but that's kind of independent almost of the therapy that you're working on. And so understanding that the primary roadblock to even getting things into the clinic, much less into the commercial marketplace, was going to be figuring out how to make these things in a cost-effective way where you can repeatedly figure out, you can make the exact same thing the exact same way with the exact same outcome every time, was going to be a massive challenge. And it wasn't going to be something we could nickel and dime our way to success for. So hence the Manufacturing Innovation Institute, funded by the Office of the Secretary of Defense Manufacturing Technology Office. BioFab's one of 9 of the manufacturing institutes sponsored by the DoD, out of 16 sponsored across the federal government. And it's the only one that's dealing, of the DoD-sponsored institutes, that's dealing specifically with medical problems. All the others are focused on more, technology-driven sorts of issues. So now we're kind of seeing some of the outcomes of the Institute, and all of those products that we funded the early development of now have the ability to take advantage of what comes from the Institute's efforts.
Thanks, Lloyd. We'll touch back for that. Now, Esther, the Assistant Vice President of JDRF, I'm going to turn to you as also a funding agency, but a funding body with a slightly different focus than the Department of Defense and the federal government.
Yeah, so my name is Esther Latres, and I'm a scientist. So my main responsibility or job at JDRF, which is the Type 1 Diabetes Foundation, is to allocate all the resources and funding to research projects, whether they're preclinical or clinical or discovery. As you all know, type 1 is an autoimmune disease. There are no cures. There's only insulin, but it's really hard to manage glucose levels from not only infants, adolescents, and adults. So the mission is to accelerate breakthroughs to both treat and cure type 1. So the way that JDRF advances the mission is in advocacy in the space of cures. For instance, 20 years ago, JDRF funded a lot of embryonic stem cell research to differentiate stem cells or embryonic stem cells into into islets at a time where there was not much funding. So this is one area we focus on regulatory, helping small companies to navigate the path of type 1 diabetes and the cures. But most importantly is also preclinical development and how can we help and accelerate preclinical development. And as you probably know, we're at a point where there are many different protocols that have been established to differentiate stem cells into an islet. Usually these stem cells can make an islet, can make beta cells and can make alpha cells as well. So it's what we call an engineered islet. And there were many protocols that were established by many different labs that there were manual protocols. So, you know, as any manual protocol, it causes variability, it causes difference from one lot to another. And I think that as we move forward and these protocols were being developed, the next thing, and it's based on our clinical proof of concept that islet transplantation can rescue or can reverse diabetes in, you know, obviously in someone who has a lot of complications from type 1. So we have the proof of concept that transplanting highly functional islets can cure or can provide a functional cure. Obviously, the challenge is that it's an allogeneic model, so they need chronic immunosuppression. So the goal of JDRF was to remove the need for chronic immunosuppression. So all these products that we developed will be a combination product of an islet, or engineered islet, with a way of protecting these cells from the immune system, whether it's we're working on obviously generating hypoimmunogenic cells with, you know, using CRISPR. We're working on encapsulating these cells. We're working on immune modulatory strategies. So at some point, the field was getting joined by engineers, immunologists, that they didn't have the capacity to make these cells and to generate islets to work in the lab. These are very complex protocols. So at some point, we decided to make an investment or a collaboration, and that's how we approached BioFab and ARMI. Like, can you make islets that are highly reproducible, that can be distributed from one laboratory to another, so the labs don't need to focus on trying to differentiate these protocols, but just on developing their own strategies on how to protect these cells. So these cells that we're working, or these islets, are they come from one of the protocols that was established in academia, and we're in the process of generating enough cells that can be distributed to many different labs. So we're quite excited about the opportunity to have a big advancement because the key is now how do you protect these cells from autoimmunity and allogeneic rejection. So that's the main focus, to move these therapies into everyone who has type 1 diabetes.
Wow. I remember Edmonton Protocol, what a stir that made 22 years ago now, I guess.
2000.
And sort of proof of concept that you could actually do that, but with using source allogeneic organs, that's never going to scale, right? But it was a proof of concept that, in fact, you could get an immunosuppression protocol that worked.
Yeah.
And I think you guys helped fund that as well.
Yes. All the clinical work on islet transplantation, although the beginning of the work on embryonic stem cell differentiation into islets. So our focus is obviously to make these therapies available and provide a cure for
Thank you.
type 1.
So, last but certainly not least, I turned to Carter Cliff of Vascugen today. I have to always check because Carter is not a funding agency and is really one of those individuals that is working on moving multiple products over the time I've known you through that valley of death and trying to figure out how to deal with both scale and funding and whatnot. So, Carter.
Yeah, thanks, Richard. It's great to be here, and thanks for being on the panel. I guess by way of background, my contribution to this field has been to try to direct iPSC-based cell replacement therapies. Some of the early ones that I worked on were with CDI and then Fujifilm after they acquired CDI. And that, for example, was a company we spun out called Opsis Therapeutics, developing iPS-based photoreceptors and RPE for transplant, and that program was partnered with Bayer. So that one, fortunately, does have the wherewithal to move forward. But I think there must be at least 100 different cell types that are potentially manufacturable with therapeutic value, and rightfully so. There's a subset of them that are the focus in diabetes and CNS and immuno-onc. But what I'm working on right now are vascular progenitors, the objective of which is to deliver them into a tissue for microvascular repair of a hypoxic disease condition. And certainly there are a number of things over the years that I've observed with respect to scale. I think that, at least for what I'm doing, the principle of beginning with an iPSC source and seeking to identify a target cell that has a high probability of being a potent cell replacement therapy or cell therapy, I think that reduces some of the risk with respect to the need for scaling later. And that's one aspect of it. But I guess for me, identifying the right cell first and then figuring out how to scale it is kind of the order of operations. And I think that's been a little bit different in the past, where you try to figure out which cells you can scale and then find out what therapeutic value they may have. So I think that the field is changing, and we're still in sort of the early stage side of that.
Thanks, Carter. We're going to begin to have some discussion amongst people now that you know who we all are. And there is a box somewhere, I know it was here this morning with a microphone in the audience, but also feel free to, Jancy's get it. Can I, Jared, can I put you in? Jancy's going to hold you the box, and Jared will go around and Jared is one of our staff in our membership part of the organization, and he didn't know he was just going to be pressed into duty. And I don't think we have utilities to take questions from the hybrid this morning, although this is available, and I'm sure that people would be happy to talk to people in general about this. So I have a kind of a question. How do you define scale? I was just thinking about it as I was hearing you guys do your introductions. There may be different definitions of scale. And I know that when I think of scale from where I sit in BioFab, that is something that everyone would be able to have this in their regional hospital across the country and the world. But from where you sit in a perspective, that may be, although ambitious, not to get to the next step in terms of scale. And it might not even be an appropriate definition. So, thoughts on what scale would mean from where you're sitting?
Well, I'm happy to start with that one. So one of the things that has typically set Army medical needs apart from the commercial marketplace is the number of patients that may be experiencing a particular issue. Although there are industrial accidents and car accidents that can have extreme consequences for those individuals, the absolute number of patients who have those sorts of injuries is not necessarily enormous. And so I'm just going to use as an example volumetric muscle loss. That is obviously a problem that affects both civilians as well as military, but the rate of those injuries in military populations when there's active conflict is considerably higher. But that also highlights an issue of how the DoD and its needs impacts the commercial marketplace in a broader way, because our need is typically cyclical. You know, for example, with the height of the Iraq and Afghanistan wars, there were lots of injured soldiers coming back with extreme injuries to the extremities. And so that represented a market for commercial entities to provide a solution for. However, if they were relying solely on us to fund their development and their eventual success in the marketplace, they would now be out of business. Just simply because the number of soldiers who are in need of those therapies is so much reduced at this time. And then at the time in the future, when some other conflict, which we can all imagine now thanks to what's going on in Europe, we would need those therapies again, and the company would not be there. So that was always a situation that we wanted to make sure that we have companies that have both a military relevance and a commercial relevance. And the scale for that needs to be adjustable. So if at some points in time, the actual amount of material that may be needed could be substantially less, and it may never have a huge commercial marketplace compared to what the DoD may want it for on a periodic basis. So it's really a right scaling more than necessarily an upscaling to massive amounts for some of these indications.
Okay, so a couple of things. I think the first one is at what point you scale. I think understanding, at least for the type 1 and for islets, I think having a good product, a good differentiation product, you understand how it responds to glucose, how much insulin it makes, what is the clinically meaningful dose. And I think we have some data, very exciting data from Vertex that has, you know, the first proof of concept of stem cell derived these islets that can rescue diabetes from at least one patient that had type 1 for a long time. So I think understanding the product, the quality, and the clinical dose is very important. And then scaling for me means we looked at the roadmap, at a clinical roadmap. Now the way the islets are used is for a very specific population, which is the hypoglycemia awareness, that they cannot manage, or they're not able to manage their glucose levels with insulin. So this is one population that's very small in the first, which is the islet transplantation. But then scaling means how do you go from one patient population in your roadmap to the next one. Maybe once you remove the need for chronic immunosuppression, you can move to the next patient population, which you will require a very different level of scaling. So I think understanding the population and your product, and I think that is key as we move through the roadmap.
So in your initial, you mentioned scaling for non-clinical studies and the preclinical studies and whatnot. And is there a factor in terms of scaling and reproducibility across the field, to potentially make comparisons amongst labs easier to make?
I think there's been many labs, and where we talk about preclinical, we talked about mostly mice. Sometimes they're mini pigs, and then non-human primates. So the scaling is very different than when you move to the first in-human studies. But most of these studies are done, the preclinical studies are done with well-characterized protocols, but not at the manufacturing or scale. And there's challenges, and as a funding agency, we understand the problems of scaling for a lot of for these academic labs, right? To put enough cells into a non-human primate to show that your product works. It's challenging. So I think that having, again, one of the reasons that we approach ARMI is to make sure that we can generate one product that is consistent.
Carter?
Yeah, I guess for me, I'm thinking of it in a kind of simple terms of the route of administration. If you're delivering to the CNS a cell therapy, a progenitor to address a degenerative condition, you're talking about, on the order of 10 million cells, something like that. If you're delivering a similar type of progenitor for a different purpose to intramuscular tissue, then you might need 10 times that amount. If you want to deliver your therapy by IV and it's going to reach a variety of tissues, or it's targeting a tissue in that route, then you're going to need hundreds of millions of cells, maybe billions. And so I guess as we've thought about where we're at from a scale perspective, in the lead up to a clinical test of the product, and when we think about who might be interested to partner with us around the asset or assets that we're developing, it seems to kind of bucket out or break down across route of administration. And if somebody is very excited about a cardiovascular repair and it needs to be IV, then it's just going to have to be resourced accordingly. Whereas if it's a retinal delivery of 1 million cells or 10 million cells, then the resourcing is potentially different.
So that's you talk about a difference for clinical indications, for scaling, different kinds of scaling. You also, when you talked earlier, you talked about the benefits of having iPSC technology, where you had more opportunity to scale different tissues. It was a limitation prior to iPSCs. What about moving through the developmental pathway for an individual product? Mm-hmm. So I know that one of the challenges of scale that I have seen over the last 20 years is scaling as you move to later phase trials.
Yeah, it seems like, again, just in the narrow space of where the engineered cells manufactured from iPS or ES sources are being developed, it seems like it's the progenitor cells that have the potential for engraftment and potency. And those cells, you may need fewer of them, potentially. You can't really access them in terms of donor-derived cell therapeutics. You're accessing more mature cells from the body. So I think that's a dividing line. And I'm not sure I'm answering your question, Richard, but I think that...
You're answering one of many questions
okay.
I have about how you move that. And I know that you've lived through this in multiple different phases and firms over the years, mm-hmm. and it's always challenging. So keep going.
Okay. Yeah, I think that the most important thing is to identify the right cell and the right IP to manufacture it at small scale. Typically, it seems like it's going to be a progenitor. There might be exceptions where the mature cells is better for that target indication. And then it's just a route of administration. I mean, we needed, a fairly small amount of photoreceptor progenitors for transplant, potentially, right? And a fairly small amount of ES-derived dopaneurons that are going into the Blue Rock trial and so on. And so I think that that's just one level of scale where you're very narrowly focused on the target cell and not on making billions of it necessarily yet. But, you do have to be cognizant of that along the way because, as you know, I mean, if you're going to scale it to a larger patient demographic, then you have to be able to scale it reliably. And, you're going to end up moving from small scale cell stacks into some sort of reactor at some point, and it's inevitable.
And how do you control that to know that your final product is the same thing?
Well,
I mean, it's one thing to be able to have a really big iPSC master cell bank, which mm-hmm. you can characterize. How do you know that when you change the scale of your manufacturing, that what comes out the other end, what you're consenting the patients to, is the same product that you thought it was?
Yeah,
in phase 1?
usually we ask your team that question and how to set our specs.
True. So specs, right? And how do you develop specs? What do you think about early on? Is this something that you think about at the end? Because you were in a situation where you're always somewhat resource constrained, and Esther and Lloyd and the commercial VCs are not going to be able to fund you to do everything that you might want to know as a scientist.
I was going to say that the first in human studies that we are funding and that we're working on are 5, 6 patients. So, and as I said before, it's usually a combination. So it's a cell source, and it is a strategy on how to protect the cells. We work with a lot of companies that they're developing encapsulation devices, that they're developing immune modulatory strategies, and they basically have 2 options. They can go with a stem cell product that they need, obviously, to partner with a company that's developing the stem cell product, or we're also funding xeno products. So we have products with pig islets as well. So it's not stem cell, but it's mostly companies that they have a strategy to protect the cell. Whether engineers, immunology, anything, they have the choice of partnering with a stem cell company, which is sometimes not that easy, or they can go to a first in human with a donor human islets. And that's a way of validating their own component there, whether if it's an immune modulatory that they're using, an immunotherapy, for example, to protect the cells. That's an option that they have. So and for the population that they're going, which is, as I mentioned, the hypoglycemic unawareness, there's enough islets out there, human donor islets, that they could go into first in human for 5 or 6 patients to validate their product, which is their strategy to protect these cells. And that's another scaling factor that needs to be considered.
So I'll actually add on to that in a particular way because, and I'm going to use skin just as the example because there are several products that are on the market already. And we funded some of them. So like the idea that you're able to produce these various skin substitute products, obviously there's a certain marketplace that is available for them, but to kind of go back to what I mentioned earlier about what the DoD sees as a potential need for that in the event of some sort of conflict, if there is a large-scale conflict, we're anticipating that it's going to be with someone like Russia and not with someone like Afghanistan, some country like Afghanistan. And the sheer number of casualties that may be resulting from those sort of weapons, such as thermobaric weapons or radiological weapons, is going to increase the volume of need. And can those companies that have these products and the way they're producing them right now scale up to produce enough of them? And probably not is the answer. So ultimately, all of it has to come back to we can't just add more people to produce more of these products. It has to be something that's more automated.
Hence why a lot of the first 5 years of BioFab has concentrated on enabling technologies. And I think we've funded 50-something from project calls based on our technology roadmap, things that we heard from people like Esther, like Carter. Yeah, we listened to the DoD as well. But people other than the DoD to say, what are the challenges in making something forward? And I mean, a lot of those are questions of where do you put sensors? Sort of the question I was kind of asking you, Carter, is how do you know when you go from a cell stack to a bioreactor that the cells aren't seeing a different environment? Mm-hmm. Not to be too anthropomorphic, but they sometimes remind me of toddlers And they say, I need more. Not very specific. And certainly, when I was at the FDA, the technology to answer those questions was rudimentary at best. And I think that that's a risk for scale for people like you. I mean, it's one thing for Lloyd and Esther, they may not be able to succeed where they are, but they're not going to have your firm go under.
Right. Yeah, I think in the case of the vascular cells, we're focused right now on scaling to support phase 1 and the supporting pivotal studies for that. I think if a company has the appropriate partners for, let's say, different routes of administration or fields of use that require larger scale, then you can start doing that in parallel. And it seems to me that you have your specs, but you've also got your FIO sort of detailed analyses, like doing RNA-seq on the populations and understanding what is changing when you test this new bioreactor from one company or another. And I did have the experience, not directly because I wasn't the one doing the experiments, but with the retinal cells, we did make parallel efforts to translate the target cell manufacturing from smaller scale, proof of concept scale, to something that was in bioreactors, and it takes time, to make sure that the product aligns. Other processes, I think it depends on the cell, because some other cell types seem to be easier to scale. So different types of non-retinal neurons, for example, it seems that there are ways of scaling those reliably without having to resort, for example, to sorting steps or other kind of steps that complicate the scaling process. So I guess for me, as long as you have something that's giving you a detailed FIO readout like RNA-seq, that at least you can kind of track what's happening on some level when you transition. But you do have to be resourced for it. Otherwise, you can't do the 100,000 per bioreactor experiment.
Right.
So that might be a whole separate problem, is how to actually enable groups to scale. Is tied into the cost of the input materials that are needed to test that question.
So I'm going to try to address something that you mentioned before, which is non-conventional regulatory approaches, indirectly by saying you asked my group what to do. But I want to, at this point, we've got maybe 20-ish minutes. If people want to ask questions in the audience, the four of us can do this for hours, and we often do. And sometimes that's where the best ideas come from. So think about your question. Jared's going to get up and he'll pass you the microphone, and folks will take questions. Non-conventional regulatory approaches, that's a challenge, being a former regulator. Right? Regulators like definitive answers. They don't necessarily like to give them. They like the sponsors to give them the definitive answers. The answers regulators tend to give is, it depends. Particularly if they're talking from a podium. But it depends on the specific details. So if I were talking to a volumetric muscle loss IND person or a skin person, I would could be a very different answer to either of those, different than I would from either an allo islet person or an allogeneic islet person, engineered islet-like cluster individual, and I would talk to Vascugen or immunology. But I think the key thing to be non-conventional is there really needs to be a back and forth discussion, and not having regulatory on the industry side of the table, but at the table. and discussing it. I was, the documentary of Medicine and Miracles about Emily Whitehead and CAR T is going around the film festival circuit right now. I happened to see it recently, and there are lots of situations in that story, which I'm not going to tell. They're still trying to deal with how do you scale this great thing to make an industry. That's a whole nother level of scaling question. But there were situations along the way there where investigators ask the agency, can we do this or that? And of course, the initial answer was, it depends. It depends on what data you present, right? And it's a feedback of how to provide data to support a non-traditional, non-conventional regulatory approach. There's a lot of room for non-conventionality in there, but the agency needs data to feel like moving forward.
Yeah, and I think everyone here knows that as your product moves through the development pipeline, the cost increases exponentially. So when we go into late development programs from either companies, small companies, or academics, is what are the experiments that we should do to show that we can move into a first in humans. So I think, obviously, we're very interested and we are very involved in this movement from late preclinical to first in humans, and we talk to sponsors. I think we want to know what feedback they get from FDA, that it's consistent, that they get what they need. But I think that's the main question. It's just this, I think this communication at some point that, okay, I've done my experiments in mice, I've done some experiments in guinea pigs, do I need to do non-human primates? Do I need to expand, the scale is costly, the animals are costly, it increases a lot. So I think as a funding organization, we're very interested and we follow very closely these conversations that sponsors have with regulatory agencies.
And you make what you hear both from sponsors and from patients known to the agency periodically in various forms.
Yeah, I mean, we hear from sponsors. They're very open at providing us feedback that they get. Obviously, from patients as well. We know that they want a cure. We, it's and obviously we're not on all in cures, but we also have all the insulin pumps, continuous glucose monitoring, closed loop systems that JDRF has made a great effort in that, right? So that's a huge voice from the patients that they tell us what are the risks that they're willing to take, right? There's a big discussion on moving from chronic immunosuppression to chronic immunosuppression free, in order, I was saying before, to get to the next population segment, right? So what are the risks that they're willing to take? They have to take the risks from their insulin pumps, from their closed loop systems, from the managing, from psychosocial effects that they have managing type 1. So what are the risks they're willing to take? And I think that's very important to hear.
Questions from the audience? Jared, right in front of you.
Morning. Matthew Doody, Cell and Gene Therapy Catapult. So the title of the session is sort of in the zeitgeist, I think, of where we are with what's going on. It's movement from niche to higher prevalence, if you like. But I've heard lots of talk about the cost of the trial work, But to what extent does the cost of goods of the actual product feature into your decision making at this point as you're going forward?
So let me start with that from BioFab, which on a good day we think that we're the UK analog, the US analog to UK catapult. Cost of goods and scaling is one of the things that gets us up in the morning. About 60% of our members are industry. The majority of the tissue engineering, regenerative medicine therapy companies like Vascugen are relatively small. But our larger corporate members are all involved in automation and manufacturing equipment of various kinds and supply of source materials. So we really think that automating the scaling, and to do that you need the sensors to know what the cells are doing so that you can make decisions about manufacturing steps around unit operations and don't rely on just in testing. And since I've got the soapbox, this idea for biologics that the product is the process really bugs me. It works really well if the product is packed red cells and you're going from one vein to another vein to ask it to do the same thing it was doing in the donor. And what you have to do is to keep it cool for 42 days. Don't contaminate it, and know what blood types are and match those up. Other than that, I think it's an excuse for not asking the difficult questions. And I don't think we can ever scale if we don't ask those difficult questions at unit operations and develop systems to look at that. I know that Carter is doing that a lot in his firms. DOD's funding us, as I said, for our technology projects to do that. And it's something I know that JDRF spends a lot of effort looking to. You've got the box back, so.
No, I was just going to completely agree with you on the process is not the product comment. I think whenever anybody falls back on that, which they do a lot less, 10 years ago that was the mantra.
Right.
But now, if we were still saying that, it would be a massive admission at failure by the industry, I think, if we were.
And to triple down on that, I would say that is something that recombinants and monoclonals 25 years ago maybe did too much when they were beginning to take off.
Ned Gordon, ARMI. First, I'd like to say thank you to the panel for sharing your views and to Dr. McFarland for facilitating this panel session. We and you shared your thoughts around scale, whether it might be the number of patients and the indication, or the route of administration and the dosage, or a little bit we've touched on decentralized versus centralized manufacturing. The question I have gets to how do you think about the regulatory approach as it relates to autologous versus allogeneic therapies? And in your experiences, maybe working with both types, have you seen similar or different approaches from a regulatory perspective?
Can I start? Okay. So JDRF has limited funding. So in some, I think, since so there is protection against autoimmunity and allogeneic rejection. We went through universal off-the-shelf products. Still don't understand if you do or if you do an autologous transplant, someone that has autoimmunity, would these cells be survived? Would it still need some level of protection. That's something that a lot of people bring up, and we should definitely look into that. I think we approach also ARMI to where the protocols on autologous iPSCs are generate us, but I think we need to understand how the cells will respond when you transplant into someone with autoimmunity. How are these cells being recognized and accepted?
Also, autologous benefits from a century of transplant science, from transfusions to hematopoietic stem cell transplants, and so I think that makes it much clearer how for those programs how they would advance into the clinic and ultimately register a product. Whereas gene therapies, introducing viral capsids and synthetic manufactured cells is a completely different effort. It's just that on the tail end of that, it might be highly advantageous with respect to off-the-shelf medicine. And so I don't know, I guess it ties back to your other regulatory question, which is what kind of flexibility can the agency give? I feel like the Japanese agency made an interesting decision to sort of open the door to having a registered product after safety, but then it's challenging there to overcome the CMC requirements. So it would seem like something that benefits from the more feasible CMC path in the US and the faster path to registration in Japan would be really great for the field. Not optimistic, but
A hybrid
yeah.
A hybrid regulatory approach.
Mm-hmm.
So, to ask you a question, what do you think, if you let something be marketed based on safety, that's a large burden for the regulatory agency to accept. Any ideas about ways that might be made more palatable?
Maybe limit it to areas in which there's underrepresentation of therapeutic assets in development relative to the severity of the population. I mean, to me, that's cardiovascular. You don't necessarily have to change the rules for immuno-onc because a lot of progress is being made there, but how much progress is being made in certain other areas is maybe comparatively limited, and the unmet need is large, and a lot of people are dying, so maybe it's worth something that's selective like that.
Thinking about doing it for a clinical entity.
No, I fully agree. I mean, obviously type 1 diabetes has insulin, so the regulatory path is...
Which is imperfect treatment.
It's like insulin, there's insulin, but insulin is not a cure. So when we have products out there, we are under the umbrella of that there's insulin. So we need to make that regulatory agencies understand that that's still insulin. It's not a cure, but we have to back it up with, the benefits of a cure.
But there's also the issue, just to play devil's advocate a little bit.
Oh, go ahead.
That you're asking, you have to assess the risk tolerance of the population as a whole, and I don't mean just of those who are going to be receiving the treatment, because you can envision numerous scenarios in which you loosen up the requirements for these various products, and then if something does go wrong, how tolerant is the population going to be of saying, well, we decided to take this risk and now people were harmed in some way. And so it always comes down to, if you can, the more data you can bring up front, the more you can justify that it was a worthwhile risk. And kind of going back to making the FDA comfortable with some of these things, obviously it's a data question and how much information can you give them, but it also, you run the risk of just vomiting data on them and suddenly they're overwhelmed with stuff, some of which may not be relevant, so all of that has to be taken into consideration as well.
And in some ways, when you vomit data on the FDA,
You run other risks.
you have other risk because it's based in scientists and engineers. And they will generate additional hypotheses based on those data and potentially ask questions that might slow things down.
Yeah.
I'm not saying hold stuff away from the FDA, but you have to know how to present that narrative, right? And some of the more successful programs that I was involved with in 17 years I was there, were things where initially it seemed rather amazing that the sponsor was proposing this. It's like, what scientific fiction book did you read? You know? And they were based on long-term conversations, back and forth, or data to something that became an overnight success in 15 years. And partially because it happens behind the veil at FDA. But that is to protect people like Carter and the funders and the scientists that JDRF funds, the companies they fund, because although it would, in some sense, it's great if everyone shared their secrets. That's really not a way to make Carter's investors happy. Right? Because of between a rock and a hard place in some cases.
Mm-hmm.
You know, you have financial realities about scaling and need to make decisions based on science and dollars.
Yeah, it would be ideal to be able to partner out fields of use, maybe routes of administration, with groups, and then sort of tailor your development to that particular partner and therapeutic application. I guess so that the other side of the regulatory path for advanced medicines being more constricted is that you end up with more, unregulated clinics and lots of other stuff because the unmet need is still there, and so people will try anything. So I think maybe there's some balance on that.
Between compliance and, the compliance on unregulated clinics, that's another topic entirely.
Yeah.
But their existence makes it more difficult for people who are trying to legitimately scale products, because it's hard to differentiate something that's in a clinical trial when somewhere on the road between Madison and Milwaukee there's a strip mall with Sam's stem cell clinic, cures everything. All right, we are getting close to the end of time. Any more questions from the audience? Okay. Well, I'm going to do something that my colleagues know I seldom do. I'm going to end a meeting on time. I started a meeting on time. I ended a meeting on time. But I'd really like to thank our panelists, because I think what you've gotten is a sense from different viewpoints of some of the challenges and opportunities, I think, for scaling. So thank you, guys.